Bismillahirrahmanirrahim. 9700 A-Level Biology, June 23, question paper 1-2, and this is the second video which explains the rest of the half of the paper. Question 21 to 40. Question number 21. How many copies of each DNA molecule will be found in a cell at the start of the stages of the mitotic cell cycle? The first stage they're talking about is G1 of interphase. Now, you know, in S phase of interphase, the DNA replication occurs and this one chromosome becomes two chromatids. Now, one chromosome is one DNA molecule. You see the double helix, ATGC, this is one DNA molecule. So this was one DNA molecule, this one. In S phase of interphase, now they become two. But then they ask you a very interesting question. They said, what will be found in the cell at the start of the stages of the mitotic cell cycle? So start of G1 of interphase, there'll be one. This won't be present. There'll be just one molecule of DNA. But it says start of cytokinesis. Start of cytokinesis means it was telophase. So one cell had two nuclei. In telophase, this one, this phase is telophase. And when it enters cytokinesis, then the cytoplasm will divide. So one cell had two nucleuses. So there will be two molecules of DNA. So that is why the answer is B. Please understand that. It says at the stages start, and the start is being made very, very prominent so that you can remember the stages of the mitotic cell cycle shown. And the cell cycle is G1 of interphase and cytokinesis. So at the start of cytokinesis, then telophase, the cell will have two nuclei and the cell will be, then the cytoplasm will be dividing and then two cells will be formed. So start of, so that was the really catch question. Then question number 22, one characteristic of DNA that's a universal genetic code. Universal means, like we say, ATP is a universal energy currency. It's a very typical word that we use in biology. Universal means in all living organisms, whether they be a lizard or it's a human being or it's a frog or it's a cockroach or it's a bacteria in yogurt or is it a mushroom or is it yeast cell in your pizza. So what is meant by the universal genetic code? Now the answer is A. All living organisms use the same triplet code for amino acids. Like AAT codes for amino acid glycine. So it codes for glycine in every organism, whether it's a lizard or whether it's a fly or whether it's an oak tree. Okay, why is B, C, and D wrong? And why would any student really answer that? B is wrong because it says all DNA triplets code for a different amino acid. No, AAT codes for glycine, then it always codes for glycine. Not all DNA triplets code for an amino acid. But that's not the meaning of universal genetic code. That is correct. I'm not saying it's wrong. Because there are some which code for a stop codon, stop and for a start. But what is meant by universal genetic code? This is correct. This is not wrong. But it's not what the answer to the question that they're asking you. All living organisms contain the same 4 nucleic acids. Actually, there are 5 nucleic acids, DNA and mRNA. But 4 nucleic acids kaise hoga? There are only 2 nucleic acids, DNA and RNA. The RNA is then of 3 different types, ribosomal RNA, mRNA and tRNA. So that is why, but plus that also does not answer the fact that what is a universal genetic code. I'm asking you, where do you get the best burger? And you say, miss, you can get mithai from whatever place, but sweet house or any sweet house. So they're asking you what and what are you answering. So there you go. Read the question carefully, read it twice, and then look at the answers and again read the question. Question number 23. Which statement about mRNA is correct? First, get this right. mRNA is single-stranded. Whether it's the mRNA or the ribosomal RNA or the tRNA, it's all single-stranded, but it folds up upon itself. Like the tRNA folds up and forms a clover leaf pattern like this. mRNA is like this, which I've just drawn. So it's a polymer made of nucleotides all joined with hydrogen bond. No, there is no other nucleotide here, so that there would be hydrogen bonds between this and this. So there is no such thing. It's not a double stranded. It's a single stranded. So this is wrong. Then each nucleotide contains the subunit sugar. Yes, that's correct. C is wrong. It always has an equal proportions of adenine and uracil. How can that be? There could be A, C, U once, and then G. There is no, the equal proportion is DNA. Why? Because always A pairs up with T. But there is no pairing up. There is no ATGC story. There is no AT and GC bonding. Hydrogen bonds form between the bases, but that's in a double-stranded DNA. And there is no U in the DNA. Please remember that. If I've written A as well and T as well and U as well, there's something wrong. I've got something wrong then. So if it's A here, it's got to be T here. If it's C here, it's got to be G here. C. If this is A, then this has to be T. But this would be DNA. We're talking of RNA. So this is wrong. The mRNA sequence is identical to the template strand of DNA. No. DNA has T. RNA does not have T, it has U. So A will pair with U and T will pair with A. So it is not there. So all these are wrong, and that is the reason why they are wrong. You must know why they are wrong. Okay, then coming to question number 24. The diagram shows part of the process of translation. You know, translation is mRNA to protein. What are the names of the structure labeled X, Y, and Z? So I'm sure everybody knows this. A very easy question. It's not a very difficult question. X is the anticodon. Y is the codon on the mRNA. And Z is the tRNA. Either you know it or you don't know it. Now, this is how I've solved the question number 25. The DNA triplets of genes are translated as amino acids or stop signals during protein synthesis. The table shows some of these triplets. What could be the effects of one substitution mutation in a triplet coding for tyrosine? Now, the triplet coding for tyrosines are ATA and ATG. So here is one, and here is the other one. Now I've told you how a base substitution will be resulting. So it can become ATC. The ATC is a stop signal. ACA is cysteine. ACA is cysteine. And ATC is again a stop signal, this one here. And ACG is again cysteine here. So what could be the effects? The triplet is translated as cysteine. This could be the answer. The triplet is coded as tryptophan. This is wrong. The triplet is translated as tyrosine. Why tyrosine? Because if it becomes ATG, ATA and ATG, ATA becomes ATG, then it's translated as tyrosine. This ATA can become ATG, but still translated as tyrosine. And translation stops at this triplet, stop signal. ATC becomes a stop signal. So that is why the answer is 1, 3, and 4, which is C. Please pause the video here, and you must first attempt these questions before you start looking at the video, because otherwise if you start looking at the video, you're not going to learn much. All these papers are available online. If you don't have them, you can WhatsApp me and I can give you the paper and you can do this. So you should first do the paper before and then look at the marking key. Question number 26. Which row identifies cells with plasmodesmata? Phloem sieve tube has it, companion cell has it, xylem vessel does not have any cell, plasmodesmata. So that is why the answer is B. Simply, you've got to revise plasmodesmata. You have to understand where plasmodesmata is present. What is it? It's a cytoplasmic thread connecting two plant cells. I said a cytoplasmic thread connecting two plant cells. So this is one plant cell. This is another plant cell in which we have cytoplasm. So we have this connection. This connection here in between this, there is this, and then there is this connection. And then there is this connection here. So plasmodesmata, please Google plasmodesmata, look at the images and understand that. I've also talked about it in my video on this chapter. Which statements about apoplast and symplast pathways are correct? In the apoplast pathway, water molecules move through free spaces in the cellulose cell walls of plant roots. In the symplast pathway, water molecules diffuse to the cytoplasm and plasmodesmata of cells. Water molecules traveling through plant tissue move by mass flow along the apoplast pathway. The Casparian strip blocks the symplast pathway and forces all water molecules to enter the cytoplasm of endodermis cells. Now, which one of these are correct? 1, 2, 3, 4, 1, 2, 3, 1, 2, 3, and 4. Now, it's 1, 2, and 3. But why? Why is 4 wrong? The Casparian strip blocks the apoplast pathway, it doesn't block the symplast pathway. The Casparian strip blocks the symplast pathway because the suberin is deposited in the wall of the cells. So that is why 4 is wrong. But 1, 2, and 3 are correct. All the other 3 are correct. So you have to either know it. This is a very difficult part of the syllabus, but you all don't understand it because of, not because of this chapter, but because you are unable to properly understand the English behind it. Where does water evaporate from during transpiration? From the mesophyll cell walls. Not either intercellular spaces. You see, the mesophyll cell walls have water on them. They have water on the surface. Just like you've got wet hair, so the water evaporates from your hair. Not from the towel or not from the from your clothes or anything on which you've got the water has fallen, but from your hair. So the water vapors are present on the mesophyll cell walls, and then these will evaporate from a liquid state. They will go into a gaseous state, and now they'll be called water vapors. So here they were in liquid state. Now they are vapor. Now they are gas. So we call it water here, but here we call it water vapor. So that is why they will be where it said, where does water evaporate from during transpiration? From the mesophyll cell walls. This has come in the previous paper as well. Question number 29. The diagram shows the outline of 3 xerophytic leaves of the same type in 3 different conditions, P, R, and S. Which description of the water potential of the cell in layer Y is correct? Now, this question actually really checks how well you know your water potential story. So the water potential has to be the highest here. Then it has to be less in this and very less in this. So high, less, very less. This one has to be the least. This one has to be the, because you see this has curled the most. So this has to be the least, and then this has to be less. So this is more. So arrow makes it bigger. Then this one has little, and this one is sort of in between these two. It's between this and this. So high, least, and this is less. So water potential of cells in cell layer Y. So we are only talking of the cells here, right? Now these are fully turgid, so the water potential has to be higher than R and S. That was the clue. Because all of these have lost water, that's why they've curled up. This has lost water a lot, this has lost water a little. So this has curled a little, but this has curled a lot. So this has got the higher than R and S. That was the clue in this. Then this is lower than P and S. And this is lower than P. So S is lower than P, but this is lower than P and S. This one is lower than P and S because it has lost all the water. But S is only lower than P. So you have to understand is we are talking of the cells in layer Y, and the fact that they have lost water. Layer Y is fully turgid in P. So it has really got a lot of water in it, so it is higher than R and S. Question number 30. The photomicrograph shows a transverse section of the leaf of a species of grass. The grass is specially adapted to grow in a dry habitat. You can see this is marram grass, which is a curled leaf. Please don't say folded leaf, it's a curled leaf. It's curled like this. Which row correctly explains how the features help the grass to grow in this habitat? Increased hair-like structures increase internal humidity, but the leaf shape decreases external humidity. Well, no, it doesn't decrease external humidity. Then B, decrease the hair-like structures decrease the external humidity. How can the hair-like structures decrease the external? Nothing to do with the external. The external is this side. Then the answer is C, but why is the answer C? Because you see the hair-like structures increase the internal humidity because the hair, then the water vapors are trapped here. Water vapors cannot leave it, so it increases the internal humidity. They stay in here. And the leaf shape in curl, because the fact that it is curled like this, so all the water vapor that escapes stays in here. So it increases the internal humidity as well. So we're talking of the leaf shape and we're talking of the hair-like structures. So all this is both increase in internal humidity and the leaf shape also increases the internal humidity. Now if the others are all wrong, then you can understand why they're all wrong. Now coming to question number 31, which statement supports the theory? Which statement supports the theory of active loading of sucrose into the companion cells? The answer is A. The pH decreases in the cell wall of the companion cells compared with the cytoplasm. Now, okay, I'm going to explain it to you. Don't worry. So, this is the companion cell. This is the cell wall of the companion cell. Now, what we are doing is we are pumping hydrogen into it. Hydrogens are pumped into the cell wall. Now, when the hydrogen ions come back in, they bring with them sucrose. And I've explained this in the videos as well to you. And then the sucrose will diffuse into the sieve tube elements, not the hydrogen ions. So why is B wrong? The pH decreases in the cytoplasm of the companion cell as compared with the cell wall. No, the hydrogen ions are constantly being pumped into the cell wall. How can the pH of the cytoplasm decrease? As soon as the pH comes in, it's just pumped out into the cell wall. It's just like an irritating student. He walks into the class and I say, okay, go out of the class. And then he tried to again convince me and says sorry and all that. And I said, no, go it out of the class. So you have to go. The H ions are constantly thrown out into the cell wall. The pH decreases in the companion cell and the sieve tube elements. No, the hydrogens do not go into the sieve tube elements. It's only the sucrose which goes into the sieve tube elements. So that is wrong. The pH decreases in the sieve tube elements compared with the companion cells. No, the pH of the sieve tube remains the same. Only the hydrogen ions are sort of coming here, then they are being pumped into the cell wall, then they are again coming back in, then again into the cell wall. So that's a continuous story is in the companion cell. Then question number 32. Which properties of water are essential for its role in the transport of blood in mammals? Now, it can't be high latent heat of vaporization. So, it has to be high latent heat of vaporization cannot be possible. We're not using, the water is just transporting. The fluid plasma is mainly water and it's just transporting it. This is transporting what? Solvent for polar substances, so glucose. So, the answer is B. It is a solvent for polar substances. That is the properties of water that we are using in the majority of the plasma is water. Because it has to transport all these. It has to transport hormones. It has to transport urea. Urea is soluble in water. We have to transport plasma proteins, large hydrophilic R groups on the outside, globular proteins. Now coming to question number 33, the diagram shows some of the events that happen between the plasma and the red blood cells in the circulatory system. So plasma, there's one which entering the red blood cells, then there is two, which is the breakdown of something which is H2CO3 to H. You see, H combines with hemoglobin, so HCO3 negative will be 2 and 4 will be chloride ions. So one has to be CO2 plus H2O gives you carbon dioxide plus water combines to give you carbonic acid. So that is why the answer to this is B. So one is carbon dioxide reacts with water inside the red blood cell under the enzyme carbonic anhydrase and forms H2CO3 which dissociates into hydrogen ions which combine with hemoglobin to form hemoglobinic acid. HCO3 negative ions move out, diffuse out of the red blood cell, and chloride ions move in to maintain the chloride shift. and to keep the electrical neutrality of the cell. Question number 34. The diagram shows the valves in the heart when viewed in cross section from above at different stages in the cardiac cycle. These are the semilunar valves, these are the atrioventricular valves. Now in diagram 2, what is happening is that the atrioventricular valves are closed and the semilunar valves are open. So that means it has to be ventricular systole, because when the ventricles contract, then the blood flows into the aorta and the pulmonary artery. So the semilunar valves are open here in this situation. You can see the semilunar valves are open here in this one and this one. While in this situation, in the other situation, we have the semilunar valves are actually closed. In this, the semilunar valves are closed. This is closed and this is closed. Both are closed. So that means this has to be diastole, relaxing, because the blood from the atria is going into the ventricles and semilunar not open. So there's that time frame which you've got to understand. Atrial contraction, then ventricular filling, and then ventricular contraction, and then ventricular emptying. So that is why the answer is A. Now diastole, diagram 1 is diastole. Diastole means relaxing, and diagram 2 is ventricular systole. Systole means contraction. So remember S with C. Systole means contraction. And this is not systole. It's systole. You can easily put it in the pronunciation and hear it to see if you are pronouncing it the correct way. Question number 35. Where are squamous epithelial cell found in the human gas exchange system? Squamous epitheliums are the ones which are found in the alveoli. So the answer is D for that, because it can be not present in the trachea and the bronchus and it's only in the alveolus. Squamous epithelium are those flattened cells of which the alveoli are made, and there's a very typical name that we use for it. Just like in the lining of the arteries is called endothelium. So squamous epithelium cells are very much in the alveoli, the thin one cell thick wall which allows the diffusion of gases out of it into the capillaries surrounding it. Question number 36. Now, question number 36, which statement about gas exchange between air in the alveoli and blood in the pulmonary capillaries is correct? So, we are comparing to the air in the alveoli and then the capillaries outside. We are comparing it to the blood in the pulmonary capillaries outside. So, this is the air in the alveolus and then this is the blood in the capillaries. And this is the capillary surrounding it. The oxygen concentration in the capillaries leaving the pulmonary artery is higher than the oxygen concentration in the alveoli. Gases must diffuse across the endothelium of the pulmonary capillaries and the endothelium of the alveoli. This is wrong. Endothelium of the alveoli is wrong. Endothelium is only in the capillaries. The elastic fibers in the alveoli walls allow the alveoli to expand to increase the surface area available for diffusion into the pulmonary. Yes, that's the correct answer. Breathing out reduces the carbon dioxide concentration. How can breathing out reduce the gradient between the blood in the capillaries and the air in the alveoli? The air is refreshed, so less carbon dioxide. So reduces between the blood in the pulmonary capillaries and the air. So how can it reduce that? In fact, when the air is refreshed, all the carbon dioxide is removed. So we have to read this question again. It's a matter of a bit of English, not very biology, I'm sorry to say. I don't agree with Cambridge on that, but still, what can we do? Beggars can't be choosers. Question number 37. The average thickness for some structures of the human respiratory system are shown. Structure human cell membrane 5 nanometers, cytoplasm of alveolar cell walls, cells, alveolar wall cells 190, cytoplasm of capillary wall cell 90, and tissue between alveolar wall and capillary wall. So I've shown you this as the alveoli. So I've shown you how I had actually, there are 5 cell membranes that is going to cross. One is this cell membrane, then this cell membrane. This is the capillary wall cell, the endothelium, and this is one, and then this, and then the fifth one is this. So 5 5s are 25. This I've written it here. Then it will pass to the cytoplasm of the alveolar wall cell. Now this is the cytoplasm of the alveolar wall cell. So that's 190. Then cytoplasm of the capillary wall cell, which is this, which is again 90 here. And then 300 is the tissue between the alveolar wall and the capillary. So this tissue here between the alveolar wall and the capillary is 300 nanometers. So I've added that, that's 605 nanometers. That's why the answer is C, which is 605 nanometers. Now I'm going to just revise this with you, and then you can see where you were going to get wrong if you were going to get this question wrong. Now you can see this is the alveolar, I'll use the yellow here. This is the alveolar cell, which are the squamous epithelium cells of the alveoli. And then you can see, I'm going to just use another color. These are the cells of the endothelium. And then, of course, you can see this is the red blood cell, which I'm going to show you with a black marker. So from the alveoli, the oxygen has to cross the one cell thick layer of the alveolus, which is this one. And then it has to cross the one cell thick layer of the endothelium of the capillary, and then it has to enter the red blood cell. So it is 1, 2, 3, 4, and fifth is the membrane of the red blood cell. So 5 membranes, so 5 5s are 25, and then the cytoplasm of this, and then the cytoplasm of this, and then the area between the tissue between the alveolar wall, and that has also been mentioned in the MCQ. So I hope you understand this, and I hope you can just revise this chapter and then have a look at it again. Then coming to question number 38, the statement refers to the disease tuberculosis. The pathogen is not accessible to the immune system. The bacterial pathogen reproduces slowly. The pathogen is not very sensitive to antibiotics. Which statement explains why antibiotic treatment for TB takes such a long time? We have to take the medicine for 6 to 9 months. So 3 has to be there. The pathogen is not very sensitive to antibiotics. That's why you take it for such a long time. So the answer is D, 2 and 3. The bacterial pathogen reproduces very slowly and not very sensitive. Not accessing to the immune system, that doesn't make any sense. Why would you take antibiotics for that? Why do you have to take antibiotics for 6 months? Normally you take it only for 5 days. If you have any bacterial infection, you take ciproxin or Azomax or vibramycin or erythromycin for 5 days, maximum 7 days. Well, why do you take this for 6 months? So the bacterial pathogen reproduces slowly and the pathogen is not very sensitive to antibiotics. Now, question number 39, the average size of some pathogens are shown. HIV particle is only 100 nanometer, Mycobacterium is 3000 nanometer, and the Plasmodium is 20,000 nanometer, and the Vibrio cholera is 1500 nanometers. Now, one type of air filter. Now, this is a very interesting question. When I read it, I got it wrong the first time as well because I realized how I was reading it wrong. One type of air filter has shown to be effective at preventing any pathogens of 1 micrometer or larger from entering the air system of a room. That's very interesting. Based on their size and mode of transmission, which diseases would the air filter prevent from entering the air system? First of all, which one is an airborne disease? HIV is not passed through the air. HIV is transmitted through sexual intercourse or through contaminated needles. It's only tuberculosis which is an airborne infection, airborne droplet infection. Plasmodium vector is malaria. Vibrio cholera is food and waterborne. So that is why the answer is D. The only disease it's going to prevent is tuberculosis. It's a very nice question. I mean, I also read it, I was confused about it when I read it the first time, and then I thought, oh my God, what's going to be the answer? A successful vaccination program provides a level of immunity where the majority of population is protected. There are several factors that affect the success of a vaccination program. Which row correctly shows the factors that affect the success of a... So the answer is B. I'm sorry, I'm running out of time. The reason is that because vaccination from 8 weeks old would of course mean that you need to fact. So this was not a very good answer. So that's why this one should have been there. Then the frequent mutation of the pathogen. Well, frequent mutation of the pathogen is not going to affect the success of the pathogen. So frequent mutations would result in the vaccination program failing. pathogen able to invade this thing again would make it not a very success story, and booster vaccination needed frequently is again not a very success story. So which can affect the success? So we have to see which can affect the success of the vaccination program. Okay, thank you very much.